Hello everyone. Thank you very much for taking the time to join me today. My name is Haley, and I am a scientist working at CN Bio Innovations. So I'm really excited to be here today to be talking about what microphysiological systems is, their applications in research, and also their potential in modernizing the drug discovery landscape. So here is an overview on today's talk. First of all, I'll be giving an introduction into drug discovery landscape and outline the challenges of our current research methodologies. I'll then go on to explain the concepts of a microphysiological system and how this could potentially bring innovation to the preclinical drug discovery landscape. And finally, I'll be introducing the MPS models we have developed here at CN Bio and the application areas. So to start off, the drug discovery process consists of multiple steps ranging from the initial target validation and compound screening steps to the preclinical test in cells and animal models before moving on to clinical trials and then finally obtaining approval for use in clinics. So while the market is constantly in a huge demand for new drugs and therapeutics, the whole drug discovery process is unfortunately very long and very expensive, also has a low chance of success. So out of the whole 10 to 15 years in drug discovery, we spend around 6 years in the preclinical phase, and that accounts for up to 33% of the total cost of the whole process. And we haven't even reached the clinical testing phase yet. And even if we reach the clinical testing trials, we still need around 8 years before the drug can be fully approved and released into the market if the testings were successful at all. But unfortunately, only 10% of all the drugs developed finally made their way to the market. So there are a few reasons accounting for drug failures in the clinical trial testing phases, with the main reason being the lack of efficacy, followed by some unexpected safety issues that could not be picked up during the preclinical testing using animal and cell models. So in preclinical studies, scientists use a wide range of tools to help them evaluate the performance of new therapeutics or molecules. So one of the most straightforward way and most traditional approach is the use of 3D cell culture and 2D cell culture and 3D cell organoid models. So they include several advantages, which is their human origin in nature and their high throughput ability. So because traditional cell lines are relatively easy and cheap to manipulate, so it can easily support high throughput research. However, they also demonstrate some limitations, which include a limited physiological relevance, because sometimes cell lines, they don't have the correct expression pattern of the metabolic enzymes and transporters that would have expressed in humans. And another limitation is their relatively low content in nature, because that would have been really challenging to set up, let's say, multi-organ model using an in vitro approach in order to allow modeling of more complex processes such as inter-organ crosstalk. So another method is in vivo approach using animal models, which allows study that is a higher content in nature because of the ability for animal models to model more complex processes such as host system interaction and inter-organ crosstalk. And of course, animal models come with a lot of limitations, with one of the most significant limitations being a low translatability to humans. It is because sometimes animals, they just have a slightly different physiological mechanisms to human, and therefore it might not be as relevant. And another limitation is the low throughput nature of animal studies, because it is very often where multiple animals will be required in one single study. And for this reason, it gives rise to associated issues such as a high research cost and also a lot of ethical considerations. And in addition to the challenges I've just mentioned, um, We are also additionally challenged with a limited access to non-human primates and also increasing pressure from regulatory bodies worldwide to reduce the use of animals from researchers. This has therefore introduced further threats to the timeline and course regarding the use of animal studies. With this in mind, it is therefore essential to consider new workflow in order to tackle the increase in demand for better preclinical research methodologies and also to address pressure from the change in regulatory landscape. So, as I've just mentioned, our research industry is currently facing increasing ethical pressure to reduce the use of animal models. So, one of the classic examples was the FDA Modernization Act, which was passed in the United States back in December 2022. So the Act has removed the mandate for the use of animal testing at the preclinical stage. Instead, it has specifically named MPS or organ on a chip technology as an example of alternative approach that can be used to demonstrate drug safety in preclinical studies instead of using animal models. Apart from that, there has also been introduction of new rules and regulations worldwide, and all of them were collectively encourage users to reduce using animal models in preclinical studies. So in order to address the limitations of our current preclinical models and also to address the pressure from different regulatory bodies over the world, there has been increasing effort to develop new approach methodologies as better alternative to animal studies. And in particular, there has been increasing evidence suggesting that the use of microphysiological systems or MPS can generate more reliable and more human relevant preclinical data. So of course, these new approach methodologies could be used in combination with the conventional in vitro cell models in order to further bridge the gap between preclinical and clinical studies. So, for those of you who are not very familiar on what a microphysiological system is, it can be known as MPS in short, or organ on a chip. They are technology that are designed to better resemble human biology, and they usually are characterized by a few common features. So, one of the key feature is the use of human cells and tissues that are normally grown in a three-dimensional structure with a geometrical confinement or patterning. So for example, we have one of our liver plate here, and this is a scaffold within the liver plate. And the scaffold sort of provides a 3D support and some structural support to the cell in order to allow the model to develop into a three-dimensional structure. And another feature includes the use of fluidic flow and environmental control within the system, which advantages include it increases oxygen and nutrient supply and also provide the appropriate stimulation signal to the cell culture. So overall, this can help the system to better mimic what happens in the human body to hopefully allow the cells and tissues to develop into a more human relevant phenotype. So here are some examples of our MPS model here at CNBio. And this technology is called the physiomimics organ on a chip technology. So this picture here shows a typical setup of our physiomimic system. So in here we have the controller which connects the system, and we can control the flow rate and different setup of the system using this touch screen over here. So this controller typically sits outside of the incubator, whereas the cells and the drivers are connected to the controller, and this part sits inside the incubator. So one example we have is the liver model, which features formation of a 3D microtissue of the liver. So this can be used for many applications related to the liver, such as drug toxicity studies and also disease modeling. And next up, we have the barrier model for applications such as the lung or the gut model. And this could be used for applications to study the transport of substances across a membrane. And finally, we have our dual organ model, which is one of our newer products, and it features linking a barrier model with a liver model. So there are interconnected flow between the two organ models, which in turn allows the user to simulate more complex processes such as inter-organ crosstalk for a wide range of applications, such as drug profiling, and I'll go through a few examples later. So while all MPS models have their own unique applications, these three are the key application areas of our physiomimics technology. So today I will focus on demonstrating how our MPS technology are predictive of human biology and how a user could easily incorporate the MPS technology into their research workflow in order to bridge the current gap between preclinical and clinical studies and of course improve effectiveness of their preclinical research. So in the following slide, I will be expanding further on how we model the human liver using our MPS technology and how it is able to resemble the structure and functionality of an actual human liver. So in order to build a human liver model in vitro, there are a couple of crucial hepatic features to consider, and that must be recapitulated in order to generate a functional liver model. So a functional human liver is usually characterized by a number of features that includes production of albumin, urea synthesis, and also expression of markers indicating different levels of hepatic damage. The important factor to consider includes the expression of various metabolic enzymes and transporters. It is because liver is a key site for metabolism, and therefore it is very important that an in vitro model will be able to resemble all these features in order to be considered functionally relevant and for use in different applications related to the liver. So in order to build the human model of the liver, first we need to seed validated cells on the 3D scaffold over here. And the 3D scaffold is collagen coated in order to allow the cells to attach to the scaffold and also provide essential growth signal for them to develop. So examples of primary cells include the primary human hepatocytes, human Kupffer cells, and human stellate cells. So these three cells are main cells in the liver, so in which I'm going to go through the function and characteristics of each of the cell type. So obviously the primary human hepatocytes are the main resident cell of the liver, whereas the Kupffer cells are resident macrophages of the liver that primarily protect the liver from different sources of infection, such as bacterial infection and also viral infection. And finally, the hepatic stellate cells are involved in the response of many sorts of liver injury. So the user has the option to either culture the main resident cell, which is the hepatocyte on their own, or they have the option to seed a combination of these three cells within the scaffold in our MPS system, depending on their specific application and specific the aim of the experiment. So the MPS liver model has been successfully characterized both functionally and structurally. So this panel of microscopy images demonstrate the development process of our MPS liver model over time and demonstrated that a 3D microtissue structure was formed within just 7 days. And after microtissue in 3D has been formed, the model also displayed a positive expression of a biochanoliculi, which is a key hepatic structure. It also demonstrates positive expression for any key structural biomarkers, which include cytoskeleton and also tight junction protein. So here are some data that were previously presented by the FDA, and it was published in literature. So, sorry, I'm just trying to figure out my mouse, sorry. Okay, there we go. So here are some data that was previously published by the FDA trying to analyze the MPS model for functionality. So they demonstrated a higher metabolic activity and also higher rate of album production than the traditional spheroid and sandwich culture. So overall, these set of results indicates that the liver model generated using the MPS technology can demonstrate an improved functionality and made this particularly relevant for any downstream applications. So moving on from the liver model, barrier model is another application of ours, which are particularly suitable to study the transport of molecules across a membrane. And I'll be going through a few examples in the following slides. So one example of our barrier model is our primary lung MPS. So in this case, primary lung cells are seeded onto transverse inserts to establish a model of the upper airway, which is the bronchial model in this case, and also the lower airway model, which is the alveolar model in this case. So again, these cells could either be cultured on their own on the apical side of the transverse, or they can be co-cultured with endothelial cells underneath and also circulating immune cells in the basolateral chamber, again, depending on the specific applications of the user. So here is our Visiomimics barrier plate, and it can fit up to 12 transwell inserts, and it's featured with a barrier flow underneath in order to allow a constant perfusion of media throughout the culture period. So upon comparison with the traditional static culture, our MPS lung model demonstrate increased relevancy to human, both genotypically and phenotypically. So we have performed some qPCR analysis on our MPS model and compared with the traditional static culture. And we demonstrate there was an increased mRNA expression for pulmonary biomarkers of specific cell populations. Examples include the AT1 cells and AT2 cells in the alveolar model, and also the goblet cells and clot cells in the bronchial models. And when we look at the histology images, it also suggested a superior tissue formation in the MPS model compared to the static control. So for example, if we look at the alveolar model over here, so the MPS model demonstrates a 3D structure containing air sacs, which is more physiologically relevant to what actually happens in the actual alveolar in human. So with this human relevancy, it therefore makes our lung MPS particularly suitable for different applications, such as applications to study pathology of respiratory infections and also disease modelings. So examples for disease modelling can include COPD and also cystic fibrosis. And as I've mentioned, since a barrier model is particularly useful to study transport of molecules across the membrane, the user will also have the option to study the properties of inhalation medicine using our barrier model and lung MPS. So moving on from single organ models, the multi-organ MPS is one of our newer concepts. And as I've previously mentioned, it can link two separate organ models together by means of fluidic flow. So in the following slides, I'll be using the gut-liver axis as an example to explain the values of using multi-organ MPS in improving the drug discovery workflow. So what's so attractive about our multi-organ MPS is that it features fluidically linking two separate organ models into one single plate, which could in turn allow a modeling of more complex biological processes, such as disease mechanisms, biodistribution of molecules, because of their ability to model inter-organ crosstalk. For this reason, the multi-organ MPS therefore introduce a potential to support studies in order to achieve an improved prediction on biological processes. Apart from that, the user will also have the option to incorporate mathematical modeling with MPS, the multi-organ MPS technology, in order to further improve translation from preclinical to clinical research in humans. So the concept of the multi-organ model is to link two models of primary human origin together. And one of most recent project is the development of a gut-liver MPS. So in this project, we collaborated with Altus Systems and incorporated their replica platform into our liver MPS model. So in short, what replica means? So replica is a product which features a primary gut model that was derived from the crypt epithelial stem cells extracted from human jejunum samples. So that is a small section within the human intestine. So it could be cultured on a transwell insert, and of course, it could fit into the transwell compartment of our dual organ plate. So for example, it could fit into the circular structure, a circular compartment in here, like a transwell insert could fit into the compartment in here and establish a fluidic linkage with our liver MPS. So by linking these two primary models together, this gut liver MPS has valuable applications such as estimating drug properties, which I'll go through later. So the replicate culture goes through two major phases, which are the expansion and differentiation phases, with each phase being characterized by a specific pattern of gene expression. And in terms of physiological relevance to human, a fully differentiated barrier of the replicate demonstrated a positive expression for intestinal biomarkers. Another advantage of using the replicate model is that it demonstrated a higher human relevancy compared to the KCO2 cell line. So what exactly is the KCO2 cell line? It is considered as the current standard for in vitro gut model, but as I've just mentioned, cell lines sometimes demonstrate limited human relevancy because of their lack of expression for key metabolic enzymes and also transporters for drugs. So when compared to KCO2, the replicate model is characterized by an improved expression of key ADME biomarkers for functionality, meaning that they might be more suitable to model the drug profiles than using the traditional KCO2 models. If we look at the physical structure of the two different models, we could see that replicate model displayed a distinct and continuous layer of mucus, which was otherwise absent in the KK2 model. So these two pieces of evidence collectively demonstrated that a replicate might be a more physiological relevant option than KK2 and was therefore more relevant for downstream studies. So after demonstrating human relevancy of the replicate model, the next step was to try integrating the replicate into our MPS model and establish a co-culture with the liver model. So we have managed to establish a special co-culture media that can that has the ability to retain functionality of both organ models in co-culture for at least 48 hours. So at the end of the 48 hours window, we performed some measurements and demonstrated that the primary gut barrier remained intact following exposure to fluidic flow and also exposure to the liver culture. And in terms of the liver culture, the functionality of the liver tissue in co-culture was comparable to the single organ control. And this could be reflected in terms of albumin production and also activity of the CYP3A4 metabolic enzymes. And finally, to validate the collective metabolic capacity of the gut and liver model, we dosed the model with a compound called 7-HC and measured its concentration in circulating media over time. So 7-HC is a compound that can be metabolized by both the gut and liver, which has been successfully demonstrated by our multi-organ model as well. So overall, this proof of concept model has therefore been validated to study the properties and metabolic profiles of more drug compounds later on in the line. So this multi-organ model has valuable applications in bioavailability profiling of drug compounds. So basically, bioavailability is a concept which measures the fraction of a drug that was administered and managed to reach the systemic circulation in the end. So for example, if you swallow a pill, bioavailability measures how much of that drug was absorbed through your gut and your digestive system and finally entering into your bloodstream. So given this open, well-designed as demonstrated in this diagram over here, the gut-liver model has the flexibility to simulate both oral and intravenous dosing of drug compounds. So for oral dosing, the drug could be added directly on top of the transwell compartment and directly onto the replica barrier. So depending on the specific transport mechanisms of that drug, the drug can be transported across the gut barrier into the circulating media underneath, which then travels to the liver tissue over here for further metabolism. And because of the multiple fluidic flow within this gut-liver MPS, as demonstrated by the arrows of different colors, this gut-liver MPS allows the modeling of the combined effect of intestinal absorption and also hepatic metabolism for drug compounds. So in terms of intravenous dosing, the drug could be added directly into the liver compartment over here and mixed with the circulation media. So this setup models the scenario where the drug was injected directly into the bloodstream and traveled to the liver for metabolism. In order to estimate the pharmacokinetic profile of the drug, the media samples can be taken at multiple time points and analyzed by LCMS in order to determine the changes in drug concentration in the circulating media over time. So here is a case study where we dosed our primary gut-liver model with a drug called midazolam. So midazolam is a drug with rapid onset that is commonly used in applications to treat conditions such as seizures and anxiety disorder. So in terms of the metabolic profiles of midazolam, so it is primarily metabolized by the enzyme CYP3A4, and there has been growing evidence from literature suggesting that the intestinal wall might be an important site for first-pass metabolism in addition to the liver. So for this reason, there is another piece of evidence suggesting why the replica primary cells might be a more suitable model for drug profiling, because we have previously analyzed and compared the properties between replicate and KCO2 and demonstrated that the replicate displayed a high expression of the CYP3A4 enzyme compared to the traditional KCO2 model. So throughout this proof of concept experiment, we completed several sets of measurements and analysis to examine the bioavailability profile of midazolam. So first of all, we have established two separate sets of gut-liver model, one with the primary replicate model and the other with co-cultured with the KCO2 cell line, and we compared these two sets of results side by side. So when just looking at the changes in the midazolam concentration in the circulating media over time, we could see there was a higher peak of concentration in the KCO2 than the replicate. And if we combine this observation with the fraction of midazolam remaining at the end of the 48-hour window, we could see that the replicate model demonstrated a higher clearance rate of the drug than the traditional KCO2 model. And we have also estimated the amount of metabolites present at the end of the 48-hour window. And in this case, it was the 1-hydroxymidazolam, which is one of the metabolites from midazolam, by estimating the relative peak area of that metabolite. So results suggested that there were more metabolites present in the replicate model when co-cultured with liver than in the KCO2 model when co-cultured with the liver. So this observation actually correlates with the level of CYP3A4 expression in the respective models. And finally, we have also carried out further calculations to estimate the oral bioavailability of a drug and then compare them with literature. So first of all, just by comparing the replicate model with the KCO2 model, so the bioavailability estimated by the model when co-cultured with replicate was much closer to the clinical data in humans. So when comparing with animal studies that were published in literature, our model also demonstrated an equivalent performance to the dog and rat models if the performance wasn't better. So overall, this proof of concept study has provided some promising results, suggesting that primary cells, when incorporated into MPS, can play a huge potential in giving a more accurate estimation on human data. So here are a few take-home points summarizing the potential of MPS technology in preclinical drug discovery. So first of all, MPS model with incorporation of primary cells has been able to model different physiological processes and conditions in multiple human organs. And for this reason, MPS demonstrates a great potential for future drug discovery studies and allows them to be more predictive in nature. And finally, a hopeful next step would be to enable drug developers and biopharmaceuticals to gain an earlier insight into the drug properties throughout their development process in order to better translate these findings into a human clinical studies. And finally, hopefully with further development, MPS model will be able to offer an in vitro solution to reduce the use of animal testing and overall improve the drug discovery landscape. And before coming to an end, I would like to give a special thanks to my colleagues here at CN Bio and also colleagues at Altus Biosystem for their support regarding setup of replicate model and the multi-organ system. So thank you everyone for your attention. There seems to be around 10-15 minutes left for Q&A. So if you have any questions, please feel free to drop them in the chat. Thank you very much. Okay, thank you. Okay. So the first question is, have you managed to move away from using FBS in your culture media? So yes, regarding the use of FBS, well, that's a good question. I'm going to put my hand up, admitting that we indeed use FBS in our culture media for some of the primary cells. So for example, in the primary liver cells, we use some FBS in the media, but at the same time, we are also looking at some human alternative, like human relevant alternative, and trying to move away completely from animal products. Thank you. Okay. The next question: how many cell types differentiate in replicate system? So the concept for the replicate system was we extract the stem cells, crypt epithelial stem cells from the crypt region in the small intestine, and they are stem cells in nature, and they will differentiate into different cell types that compose of a human gut. So I'm not sure exactly how many cell types that will differentiate into, but I am confident that it will differentiate into the cell type that is like the good number of cell type that will be able to replicate the physiological properties of the human intestine in vitro. Okay, so if there isn't any more questions, I am going to um, put the talk to an end. So if you come up with any further questions in the meantime, feel free to drop me an email. So I've dropped my email address over here. And if you're interested in exploring our range of MPS products, you are also very welcome to visit our website and give us a shout. So, well, thank you once again for your time and have a nice day and have a nice week ahead. See ya. Thank you.